All right. Thank you all for having me here. I'm excited to talk about my work, which I'm going to be focusing, by the request of the presenters, I'm going to be focusing on this paper that we just put out on bioRxiv about a month ago, which has the same title as my talk, except I threw in the word protein to be more clear for for this audience on exactly what kind of biosensors we were talking about. And so if you're interested in reading this paper, I put a link of it on this opening slide for you to check out. And so, in this audience, we're all really well aware of the types of applications that biosensors can be really valuable for, a wide range of things. In our lab, we think often about kind of threat detection, what kind of molecules might we be interested in sensing in order for national security issues, as well as metabolite sensing. I'm involved in agrochemical control of plant traits, as well as really interested in developing more spatiotemporal control of engineered cell therapies with our biosensors. But the point that I really want to emphasize here, my work also is the types of molecules that we need to be able to sense for many of these applications. And in the Whitehead group, we've been think about these as drug-like molecules. And our definition is that these are all kind of complex molecules with many rotatable bonds and multiple different functional groups. And these molecules are going to be harder to go after than simpler, more planar molecules that have often been kind of the preferred starting point for designing protein binders. And so an example of these type of drug-like molecules that are relevant now are synthetic opioids. And in particular, today I'm going to be talking about this class of synthetic opioids called nitazines. Nitazines have been emerging because illicit drug manufacturers have a lot of incentive to change up the types of molecules that they introduce into the drug supply in order to skirt bans and to get users hooked on more addictive compounds. And so these nitazines are two benzimidazole compounds. And it's similar to fentanyl as a synthetic opioid, but depending on the type of nitazine, they can be up to 40 times more potent. And so this central structure is surrounded by three different functional groups that in the literature are conventionally known as R1, R2, and R3. And depending on the different combination of functional groups, it composes a certain molecule with the name. And so one example of nitazines that have been identified in the drug supply as a particular concern is this compound isodonitazine. And so these have been a growing concern across Western Europe and the US and Canada for the last decade or so, particularly in the last couple of years. Um, and while there is a commercially available test strip to detect them, our ability to continue to track the spread of these drugs and treat patients is going to depend on having greater access and higher quality, different tools to detect them with. And so the Whitehead group has been collaborating for several years with Ian Wheeldon and Sean Cutler's groups at UC Riverside to develop the plant hormone receptor proteins PUR1 and HAB1 as a biosensor system that can be engineered to bind different small molecule ligands. So how the system works is PUR1 will first bind a small molecule ligand. In the wild type system, this is the hormone abscisic acid. And this binding induces a conformational change that enables the binding of the partner protein HAB1, which is a phosphatase. And so this chemically induced dimerization process is this two-step binding process. And so one example of how this sensor has been used was Sean's group several years ago, a great example, used a PUR1 sensor engineered to bind the agrochemical amandipropamid, expressed this in plants and used it to control drought tolerance. And so why we really have been focusing on engineering the PUR1 sensor as a biosensor is that this architecture of the sensor is really well suited to a lot of different applications. And so we think about this as being kind of general, tunable, and portable, in that it's the general sensor in that the single protein PUR1, which is generally illustrated in blue in all of these cartoons, is the only one that directly binds our ligand, um, or the main binder of our ligand. And we can engineer different variants of Pier 1 to bind to different small molecules, and they still will be sensitive to that partner Hab1. And so it's easy to engineer many different versions of Pier 1 binding different molecules. The two-step binding process results in this nice saturable isotherm that is really easily tunable to different protein concentrations and so useful for many applications. And this idea of a portable biosensor where the molecular recognition piece is separate from its signal transduction method. And so we can couple our molecular recognition piece, the two proteins, Pier and Hab, to many different output signal transduction methods in order to be able to use this sensor in many different applications. And so for many years, we've been working on engineering Pier 1 sensors to bind a wide range of different small molecule ligands for different applications, which we're able to do because this Pier 1 protein fold turns out to be really amenable to binding different small molecules. And so generally how we accomplish this is by building libraries that we can then go and screen in yeast-2 hybrid to identify lower affinity Pier 1 variants that can bind our ligand at like the micromolar level. And then through more optimization, we can get to higher affinity sensors that we can then quantify their binding affinity by yeast surface display. And so through this approach, we've engineered really dozens of novel, at this point, possibly hundreds of novel Pier 1 sensors binding a range of synthetic cannabinoids, organophosphates, and other compounds. So while our library screening process has been really successful, we wanted to start introducing computational design to help guide our libraries towards new ligands of interest that we couldn't detect in our naive comprehensive libraries. And so you can see why this starts to matter just by looking at the math of library size, where in the example that I showed before, we were screening a double mutant library. So combinations of 2 mutations among 25 to 30 positions inside the Pier 1 pocket. But we are really limited in the size of library that we can express in yeast, to the point that combining more than 2 mutations across that entire 25 position set is mathematically really challenging. And so my idea here is that we could introduce targeted diversity into these libraries, combining more mutations at fewer positions with fewer different amino acids, rather than all possible positions to all possible amino acids. And so with this approach, though, we would need information on how to tailor a library to a particular molecule. But we could express these libraries we can express these libraries, building them through Golden Gate assembly, and continuing to screen in your yeast surface display. Okay. Anyway, okay, so when we think about when we thought about how we're going to tailor a library towards a particular molecule, when we started to study the crystal structures that we had available of certain engineered biosensors, we really saw a pattern in how our ligand binds in the Pier 1 pocket. And this pattern here is that we have this hydrogen bond acceptor that is really consistently aligned in the same orientation with around a coordinated water in the same several residues on Pier 1. And so this gave us that idea that we could dock novel ligands along in the same space. So we align a new ligand along the same shared structural feature of the ligand in a solved crystal structure. So in this case, along the hydrogen bond acceptor of abscisic acid, shown in orange, in this process that we call structural replacement. And so when we first applied this idea of structural replacement to synthetic opioids, our target of interest was benzylfentanyl, which is a closely related analog to fentanyl that we can purchase in a regular academic lab. So using this modeled Pier 1 benzylfentanyl structure, and a combination of Rosetta fast design and rational design informed by our previous experimental studies of other engineered Pier 1 sensors. We selected combinations of mutations at positions designed to optimize benzophenol binding. And this library contained up to 9 mutations per variant, which was a huge increase from the earlier double mutant library we were screening, with most of these mutations to hydrophobic residues. And so we screened this library against a panel of ligands by yeast-2 hybrid. And while we actually never found a benzofentanil sensor hit by yeast-2 hybrid, this increased number of mutations per sensor variant in the new library helped us to detect our first hits for several nitazene compounds at a modest limit of detection of about 100 to 200 micromolar for most of the molecules. And so this was a great breakthrough. It's the first time we'd been able to sense nitazenes with Pier 1. But before we kind of evolved these sensors further, I really wanted to improve this computational design method in order to apply it to our future library constructions. And so there are two major changes, two major improvements that I wanted to make to this method. And so the first is relevant because these drug-like molecules that we're modeling have thousands of possible conformations of the ligand in space. And so we had hypothesized that each high affinity Pier 1 biosensor binds a single ligand conformer, and that this conformer is among the lowest energy conformations if you simulate them in solution. And we had previous data from MD simulations that supports this. This is true for several engineered Pier 1 sensors. And so we applied this idea to biosensor design by simulating ligand conformers as temperature replica exchange molecular dynamics method developed by our collaborators at CU. And using this to identify the lowest energy conformers in solution to use for our modeling. And so in this example shown here for isodonitazene, the 3 lowest energy conformers, they account for 97% of the observed conformers in solution. And so we can take only these 3 conformers forward into our modeling. And so the second point of improvement is that it was an assumption made with the structural replacement alignment method is that a new ligand will occupy the exact same space as another ligand bound in the Pier 1 pocket. And that's pretty unrealistic. Now, we know that it still coordinates the water in the same way, but there's likely to be slight variations in the exact alignment of the molecule in the pocket. And so we wanted to be able to sample a wider range of these perturbations. And so I updated our I updated our software in order to align each selected conformer in the Pier 1 pocket and then apply PyRosetta movers to iterate over many different slight perturbations of that alignment. And we can select only the conformer in alignment that doesn't clash with the Pier 1 molecule in space. And so with this method, we can start to integrate also our experimental data from these yeast-2 hybrid screenings, modeling ligand binding to actual Pier 1 sequences that bind our ligand at low affinity. And so here we're screening out alignments that clash with both the protein backbone as well as the protein side chains. And so we're able to screen against these actual sequences that we had identified by yeast-2 hybrid. And when we find ligand alignments that can satisfy these sequences, we're able to visualize how this ligand fits into that space, which is really valuable for designing more focused libraries for these particular molecules. And so I'd simulated binding of menitazene as well as isodonitazene against yeast-2 hybrid data in order to inform our next generation library tailored for these nitazenes. So we screened this updated library, again in use2 hybrid. We were able to identify tighter binding sequences to several different nitazines. And we ported a particularly noteworthy sequence into constructs for yeast surface display, as well as a split luciferase assay, in vitro assay, to validate this binding. And this sequence is able to bind both the standard nitazine as well as the variant menitazine at about 1 micromolar minimum dose. Um, and so we're also showing here that this sensor does not respond to benzylfentanyl. What is important is that we're starting to identify constructs that are specific for our nitazines over other types of synthetic opioids with different structures. So we think about what we might want to, what applications do we want to design for? And so there are two basic applications that we were really interested in. And the first is that we wanted to identify sensors that would be able to detect metabolic byproducts of these molecules. In particular, isodonitazine is known to be metabolized into this 4-hydroxynitazine. So we wanted to be able to detect that in complex matrices like urine. Um, but we also thought that the more useful sensor for example, screening the drug supply would be a pan-nitazine sensor that can recognize many different closely related nitazine variants. And so in order to kind of optimize towards these engineering goals, we built a series of libraries informed by computational design, um, and kind of screened them in this iterative process. And through each of these steps, um, really modeling kind of the previous round of data from either originally from yeast2 hybrid and then moving into data from yeast surface display once we had sensors that were tight enough binders that we could see them on the yeast surface. We're able to take that computational modeling data, design a new library with that information. We're going to sort for binders to our molecules of interest, and then we can identify which sequences are binding from deep sequencing. So in this kind of two-step process, we first were identifying higher affinity sensors. And so we started here taking that sequence that bound both nitazine and menitazine. We first started with a deep mutational scan, identifying positions that improved binding. And when you compare enrichment to both menitazine and nitazine, which differ only by this single methyl group, we start to identify these positions that improve binding for both molecules. Um, there's also kind of the side quest here is the set of mutations that you can that allow you to discriminate between the two molecules, but that was less relevant for the goals of a pan-nitazine sensor. And you'll see here that many of these positions are all kind of at closer to the top of the molecule around this nitro group that we use as the hydrogen bond acceptor in our alignments. So this first library for improving affinity was more optimized around the top of the molecule, and we were able to improve our binding affinity into the nanomolar range, and also we're able to detect this 4-hydroxynitazine molecule that's that important metabolic byproduct. And in our next step, taking that data and building into a new library that we're optimizing for the breadth of the sensor. And in this case, we're now starting to consider mostly these positions at the bottom of the pocket around these more variable R1 and R3 groups. And so the mutations that we incorporated here were really around how can we sense the largest range of different functional groups that we're likely to see in these nitazine molecules. And so we're showing here on the right here is data in yeast surface display showing binding to both multiple different R1 groups, which is kind of the functional group we had mostly been considering before, as well as this R3 group, where we see fewer changes between different nitazines, but we're also now able to recognize a wider range of structures at both positions. And so the final step of actually having a final sensor that would be relevant for clinical applications is deploying these in an assay and context that's clinically relevant. And so we're moving this to our optimized split luciferase assay, where we use an internal calibrator protein so that we can compare the ratio of ligand-dependent luminescence to our background GFP fluorescence. And so this final data is showing both our sensor optimized for binding that 4-hydroxynitazine, as well as our pan-nitazene sensor recognizing many different nitazene compounds. And really important here, we also screened against several different non-nitazene illicit drugs, and our sensors are not reacting to benzophenol as well as codeine and heroin. So we're really pleased with these final sensors, that there's specificity to nitazenes as well as their ability to detect and have a wide range of clinically relevant molecules in these clinically relevant matrices of urine. Urine in particular, I think we are also testing. Actually, we're not going to go in that direction. So we're really excited to see these potentially be developed into an actual product, and we're working on doing some commercialization through our universities. I think I'm right up against our time, and so the things that I'm kind of interested in highlighting here that are might be relevant for other people are thinking about some of the big breakthroughs for us that were really important was thinking about how we're modeling these different ligand conformers with that molecular dynamics simulation. Zeroing in on relevant conformers was really valuable for us, as well as how we're integrating our experimental data into this computational modeling, and the platform of the Pure One sensor that we're using that can easily be deployed into a wide range of different assays that have been really important for this translation to clinical applications. And so with that, I'd happily take any questions. And also, I'm currently looking for my next gig. And so if you have any suggestions, projects that you think that I would be a great fit for, or connections in industry that I would be happy for you to reach out to me.